Hi everyone, my presentation is on spot symmetry and how that predicts body condition in spotted salamanders or Ambystomata maculatum. So for distribution of spotted salamanders, they're found in eastern North America, primarily in hardwood and mixed deciduous forests. They're found everywhere in New York, and we could definitely find one in Oneonta since we have a lot of mixed deciduous forests. They're also found in Western Canada. So as far as how to recognize it in the wild, they're pretty distinctive with two lines of yellow dots spanning from the head all the way down to the tail. Being from family Ambystomatidae, they also have very prominent costal grooves, as you can see. Their body is dark dorsally with a lighter gray ventral side. For its life history, spotted salamanders don't reproduce in permanent bodies of water such as ponds or lakes. They only reproduce and lay their eggs in vernal pools, which eliminates a lot of predators such as fish or frogs. They're also fossorial, so they're found underground or rocks and logs for the majority of their life, and they only emerge in early spring when mating occurs, which involves internal fertilization with a spermatophore. Spotted salamanders are also aposematic because they have poison glands behind their head that they use if they are threatened by something. So their yellow spots aren't any sort of mimicry, they're actually have that. So the IUCN says that spotted salamanders are of least concern. Federally, they're also considered stable, but in New York, they're listed as a special concern species. They're threatened by a lot of the same things as other salamanders and amphibian species in general in New York, such as habitat degradation from housing or urban areas, roads and railroads, and also logging. Pollution is also a threat since they are so dependent on vernal pools. Acid rain can really decrease the pH of those ponds, make it really acidic, and hurt individuals, especially eggs residing in that water. So those are all threats. The main question that the authors posed was, will fluctuating asymmetry in spotted salamanders for their spots and limbs correlate to the state of individuals in terms of body condition? Even though they used museum specimens for the study, spotted salamanders are pretty abundant, so they're a good test group for the study. This, therefore, is a lab study, and everything was done in the lab. It would be pretty hard to do the study in the field because it requires the individuals to stay still and be as straight as possible. So even though this isn't the first study to do something like this study is more focused. Other studies have focused on individual quality in general, while this one focuses on developmental stress based on their environment and genetics. So to get to the methods, they used body condition for the dependent variable, and body condition is a measure of individual quality, so this is a good variable to use to see whether symmetry is correlated to individual quality. The main independent variable that they used was spot asymmetry, but they also tested spot roundness, spot size, spot number, leg symmetry. With that, they measured hind length and body area. They compared each variable with body condition to see whether there was any sort of correlation. Fluctuating asymmetry has definitely been shown to be correlated with developmental stress in different environments, so these variables are a good summation of fluctuating asymmetry. So like I said before, they used museum specimens. They dried them fully for 5 minutes, placed them on a blank background, and photographed them from above. As you can see, they had to pick specimens that were as straight as possible, so they couldn't use specimens that were warped in any way or missing limbs or anything like that. They had to see all the spots. And from that, they determined the roundness of each spot on a scale of 0 to 1, 1 being the most round, the number of spots on each side, so they counted the amount on the right and the left, and determined the area of each spot. From the photographs, they also determined the length of the hind limbs and the body area of the individual they were testing. After all of that data was collected and organized, they made linear models for body condition versus spot number, body condition versus spot roundness, body condition versus spot area, body condition versus body area, and body condition versus leg length. And they also compared spot number asymmetry and leg asymmetry. So a lot of these linear regressions showed no significant correlation between the data. However, leg asymmetry was positively correlated with spot number asymmetry, meaning that when the legs were asymmetrical, the spot number was also asymmetrical. And some other regressions that were significant was spot number asymmetry and leg length asymmetry versus body condition, which turned out to be negatively correlated. This means that when the body condition was less, the asymmetry of legs and spots were high. To clarify that more, when the individual was in bad shape, the spots and legs were also more asymmetrical. So overall, the authors concluded that individuals in this species are supposed to have symmetrical spots, since symmetrical hind lengths tended to have a symmetrical number of dots. Along with that, symmetry of spot numbers was associated with high body conditions, and so the authors said that in the future, spot numbers could be used as measures of individual quality. Some broader implications includes the fact that the environmental and genetic stress can deter bone growth in general throughout the life of the individual. Spot symmetry can therefore show individual quality and whether something in that environment was putting stress on the individual. In terms of conservation, that stress can be determined and eliminated to make sure a more favorable environment for future generations. Spot number asymmetry is easier to measure than leg symmetry. You can just count the spots rather than having to measure the legs in the field. So it might be more efficient to use that as a measure of fluctuating asymmetry in future studies. A new possible question to be posed is that if food availability is a factor of stress and that makes fluctuating asymmetry heightened, why wouldn't that symmetry just be from changing seasons and historical stress of a depletion of food for a couple months rather than in general environmental stress? So, like I said before, anything from habitat degradation or pollution in water, anything like that. Is it everything together, or is it just because of the lack of food for a couple months? And those are all my sources. Thank you.